So we have Mr. Watson and Mr. Crick, and they're developing this new DNA model, and they're successful in it. And when this happens, we kind of learn a couple new things about the DNA structure. The first thing we learn is that DNA is a right-handed double helix structure. And you might be asking, what does it mean by the right-handed structure? What does that exactly refer to? And you might have heard that with proteins as well. It's just really a reference to the double helix twist. So if we have, so imagine this is you standing right over here with your sexy ass hat, and you're looking at through the DNA. Let's say this is the DNA right over here, right? So 1 second, let me just copy this real fast. Copy, whoops. And we're just gonna paste it right here. That's plagiarism right there, but don't worry about that. So let's imagine that this is you right over here, and you're watching through the DNA double helix. So you might be you're going to notice that the DNA is twisting in a certain manner. It's either opening clockwise or it's opening anti-clockwise. Well, guess what? We just said that DNA is right-handed. We will never see a left-handed helix. But the point is, we have right-handed and left-handed protein structures as well. But it's just a reference to the twist. So we can hopefully understand that if you're standing here and looking through the DNA, you're going to notice that the DNA is twisting clockwise. It's a right-handed structure. So that's the only thing that the right hand is referring to. And I want to highlight it again that it's a clockwise rotation. The next thing we want to learn is that it's a double helix. The double helix is referring to the double helix over here. So we have one helix, we have a second helix, and that kind of also highlights that it's a double strand. But the most important thing, to me at least, in the Watson-Crick model was the parallel and the anti-parallel strand of the DNA. And what that really means is that, okay, so let's focus on this DNA right over here. So we have two possible ends in the DNA. We have the 5' end and we have the 3' end. So we have the 5' end of one template, and that's one strand of the DNA that's gonna travel towards the 3' end. The other anti-parallel is gonna be going from the 5' end to the 3' end. That's the only thing. That's what it means by the parallel and the anti-parallel. And this gives it unique properties, and when it comes to DNA replication and when it comes to, copying mRNAs and whatnot, it gives it a very distinguished pattern, and we'll discuss that in later videos. And remember that this DNA strand over here is stabilized by the van der Waals forces, and these bonds are hydrogen bonds between the bases. Another interesting fact that you should memorize at this point is that adenine and thymine will form a double bond between them, whereas a cytosine and a guanine will form a triple bond between them. And the reason why this happens is because of the structure of the adenine cytosine, the adenine nucleotide versus the thymine nucleotide. The way the interaction between the two molecules will result in a double bond. Whereas in cytosine, if we look at the structure, we're not going in detail and talking about the structure. We're just kind of recognizing why we have double and triple bonds in comparison to one another. In cytosine and guanine, we have triple bonds that are made, and these triple bonds are formed because of the molecular formula and the molecular bonding between the two nucleotides. So, of course, scientists like to weight everything. An interesting fact you probably want to memorize is that 100 base pairs is equal to 1 kilo base pair. This is how we kind of weight base pairs when it comes to chemistry. So in humans, we have 10 to the 9 base pairs, and in bacteria, we have 10 to the 6 base pairs. This is a random fact that you might be able to understand the entire weight or the base pairs, the estimated base pairs inside the human being and the bacteria. So hybridization is the annealing of two complementary strands. This is a good definition to understand. And annealing is when they come together and they're kind of being formed in a sense. And the opposite is denaturization, is when they kind of break apart. Now, here is a question to ask. If the amount of thymine is known, the amount of adenine can be calculated. So now let's say that we have a DNA structure. Let's just use the same thing, the same thing. So we were being asked that if we have thymine, we know the amount of thymine. Can the amount of adenine be calculated? Now we have thought about it. Let's think about it logically. So thymine is going to bond with adenine, and the number of thymine, as many thymine nucleotides we have, the ratio is 1:1 because one thymine is going to bond to another adenine. So this means that yes, it can be calculated. The ratio is 1:1, and it is complementary, which means that, like I said, it's a 1:1 ratio. Question number 2: if the fraction of a pyrimidine nucleotide and the double helix's molecular weight is known, the amount of adenine can be calculated. Now let's think about this logically, right? So pyrimidines and purines have a 1:1 ratio. And because of that, this really doesn't make any sense. You cannot calculate. This doesn't make any sense. You can't do that. This is not possible. Yeah, exactly. Purines and pyrimidines have the same ratio. And hopefully you can you don't need to memorize this. You just got to understand this. It's going to stick in your head. Next question to ask. A strand with more pyrimidines will bind stronger than its complementary. So we have discussed that, some bonds, cytosine and guanines will have triple bonds, whereas adenine and thymines are going to have double bonds. So does this mean that if we have more pyrimidines, there's going to be a stronger bond? There's going to be a stronger linkage between the two nucleotides if you have more of these? Well, the answer is no again. It's a 1:1 ratio, so that really doesn't make any sense. In reference to pyrimidines, exactly. Next question. So we have AT, and is it complementary to AT? Well, think about it for a second. This is a little bit confusing if you don't understand the anti-parallel concept yet. So this is just referring to, well, it is complementary, but I'm just going to explain why it is. So we have AT, right? AT. Now notice that the complementary strand or the anti-strand is going to have okay, so we have a T, we have an A, so adenine goes with thymine goes with adenine, guanine goes with cytosine, thymine goes with adenine goes with thymine, and so on. So notice the sequence: ATA, CAT. Okay, so it is complementary to it. So yes, excuse me, it is complementary. So just to really quickly summarize everything we learned, if there's something you got to take out of this video, I would say is understanding that we have parallel and anti-parallel strands inside DNA, and we pretty much know it's double-stranded, and hopefully understand what it means by right-handed DNA. Why it's right-handed? It's just a reference to the twist of the entire structure right here. And we got to memorize the fact that adenine and thymine will form double bonds, whereas cytosines and guanines are going to form triple bonds. And this is, again, justified by the structure of the nucleotides.